right, let's talk about the microscopic world of enzymes. These things are, well, they're the incredible biological engines that make life itself possible. I mean, they run reactions at just astonishing speeds. But, to really get a grip on their power, let's start with something a little more dramatic. Picture this. You're a small beetle, and something is about to eat you. What do you do? Well, nature has let's just say, an explosive answer. Okay, take a look at this guy. This is the bombardier beetle. And when it gets scared, it doesn't just, run away or hide. No way. It literally blasts its attacker with a boiling hot, disgusting chemical spray. And this isn't some simple little squirt. We're talking about a violent, high-speed chemical reaction. And the secret ingredient making it all happen? You guessed it, enzymes. So, you're probably wondering, how on earth does this little beetle pull that off? Well, inside a special chamber, it mixes two chemicals, hydrogen peroxide and hydroquinone. But here's where the magic happens. It has two specific enzymes, catalase and peroxidase, that act as total supercatalysts. They trigger this insane chain reaction that happens in a blink of an eye. And the result? Boom! That explosive boiling hot spray. It's a perfect, maybe slightly gross example of just how powerful enzymes are.
Okay, so that's a pretty wild example of what enzymes can do. But it brings us to the big question, right? What are these things? What are these molecules that can make reactions happen millions, even billions of times faster than they would on their own?
So at their very core, enzymes are what we call biological catalysts. The best way to think about them is like tiny little protein machines. Their whole job is to speed up one specific chemical reaction. And here's the really cool part. They do their job without getting used up. They just go again and again and again. Honestly, without them, life as we know it just wouldn't exist. Period.
And these little machines, they work everywhere. You've got intracellular enzymes, which, as the name suggests, operate inside our cells. They're the ones driving all those metabolic processes that keep us running. But then you've also got extracellular enzymes. These are the ones that cells kind of spit out to do jobs outside the cell. The classic example, all those digestive enzymes in your gut breaking down your lunch so you can actually use the nutrients.
Here are the core details regarding this topic. Review them intently for they are deemed crucial for your finals.
All right, so we know what they are and where they work, but how do they do it? I mean, how do they grab onto their target molecules, we call those substrates, and make the magic happen? Well, it all comes down to their incredibly specific and kind of beautiful three-dimensional shape. Okay, so the first big idea that scientists came up with was called the lock and key hypothesis. And it's a really simple, elegant idea. It says an enzyme has a special spot on it called the active site, and that active site is shaped perfectly to fit one and only one specific substrate molecule. Just like your house key only fits in your front door lock.
These are the key points related to this topic. Focus on them carefully as they are considered essential for your exams.
And this image here shows it perfectly. See? The enzyme is the lock, and that substrate is the key. When they bind together, they form this temporary thing called an enzyme-substrate complex. And right there, in that complex, that's when the reaction gets going.
It's a really good analogy, right? Super easy to understand. But it does make you wonder, are these complex, floppy protein molecules really as stiff and rigid as a metal lock? Well, it turns out the answer is no, they're not. So a better, more up-to-date model is what we call the induced fit hypothesis. The idea here is that the active site is actually pretty flexible. When the substrate gets close, the active site actually shifts its shape just a little bit to create an even more perfect, snug fit. A better analogy might be putting on a glove. It kind of molds to your hand to get that perfect fit. So let's just quickly compare those two. You've got lock and key, which says the active site is rigid and already perfectly shaped. Then you've got induced fit, which says, nope, the active site is flexible and it adjusts itself to hug the substrate perfectly. And it's that little adjustment, that little hug, that makes the whole thing so much more efficient.
Here are the core details regarding this topic. Review them intently for they are deemed crucial for your finals.
And here's a fantastic real-world example of this in action. What you're looking at is the enzyme lysozyme. You actually have this in your tears and your saliva, and you can see its substrate snuggled deep inside that active site. Lysozyme is a huge part of our immune system. Its job is to find bacteria and just obliterate their cell walls. And just look at how that enzyme molecule is wrapping around the substrate. That is a perfect picture of induced fit.
Okay, so we've got this idea of a perfect fit, this induced fit. But how does that actually make reactions go so much faster? What's the secret? Well, the answer? It's all about energy.
See, for just about any chemical reaction to get started, the substrates need a little push, a little boost of extra energy. We call this the activation energy. The best way to picture it is like you have to push a big boulder up a small hill before it can go speeding down the other side. That effort to get it to the top of the hill, that is the activation energy.
And this right here, this is the enzyme's biggest secret. Look at the top graph. That big hill, that's the activation energy a reaction has to overcome all on its own. It's a tough climb. But now, look at the bottom graph. When an enzyme shows up, it basically just flattens the hill. That activation energy barrier is lowered dramatically. The enzyme makes it so much easier for the reaction to happen, and that's why it happens so incredibly fast.
Okay, this is all great in theory, but let's see how we can actually watch this happen and measure it in a lab. And for that, we can use a classic experiment with an old friend, the same enzyme we saw in that bombardier beetle, catalase. So here's the setup. You take some catalase and you mix it with its substrate, which is hydrogen peroxide. As the enzyme gets to work, it starts breaking down the hydrogen peroxide into plain old water and oxygen. Now, since oxygen is a gas, we can just collect the bubbles. And by measuring how much gas we collect over time, we can track exactly how fast that reaction is going. And this graph shows you exactly what happens. It's plotting the total amount of oxygen we've collected against time. Now, notice right at the beginning, the line is super steep. That's when the reaction is going at full blast. But then, as time goes on, you can see it starts to level off. And that makes sense, right? It's because the enzyme is running out of substrate to work on. And that fastest part, that really steep bit right at the start, actually has a special name. We call it the initial rate of reaction. This is the moment when there's tons of substrate around, and the enzymes are just working as hard as they possibly can. It's really the best way to measure the enzyme's top speed for that particular setup. So if we look back at that graph, we can actually calculate this rate. In the first 30 seconds, the reaction produced 2.7 cubic centimeters of oxygen. So we just double that to see what it would be in a full minute, and we get an initial rate of 5.4 cubic centimeters per minute. How cool is that? We can actually put a hard number on how fast that enzyme is working. So, there you have it. Enzymes are basically life's ultimate accelerators. They lower those energy barriers and make all of life's chemistry happen at speed. But think about it. In a system as complex and delicate as a living cell, you can't just have gas pedals, right? You'd spin out of control. So that leaves us with one final, really important question. If enzymes are the accelerators, what then acts as the brakes?
The screen highlights important terms for this topic, arranged based on various exam boards. These keywords require strong focus during preparation due to their importance and must not be overlooked at all. Additionally, there is a past paper review for each board and paper category. Pay attention to this as well. It will greatly enhance your preparation. Moreover, try to understand how each keyword works in context instead of just memorizing it alone. Practicing these terms regularly in past questions will improve your accuracy and exams outcomes.
Please subscribe our channel to get more educational videos. Thank you.